Hey biology students, Professor Gideon here, and in this lecture video we are talking about complement. So when I use that word complement, different than when we use the word compliment with an I in the middle, in that I would give you a compliment, you look very nice today, I love that shirt you're wearing, that is a compliment. So different word in the connotation here of the immune system. So notice there's an E in the middle. So complement is actually in a series of proteins, and these proteins assist in the second line of defense. So we're still describing nonspecific or innate immunity, and we call this the second line of defense. Still part of innate immunity. So I like to talk about complement at this point because we've already discussed two other really important processes of the second line of defense: inflammation and phagocytosis. So I want to tie those two concepts together for you, and then I'll describe how complement acts to help. When we use that word complement, think more like help. It's helping or assisting the other mechanisms of the second line of defense. So I drew out a diagram here as a map for us to take a look at. So in a previous lecture video, the video on inflammation, we discussed what happens if somebody steps on a nail. The nail goes through the skin, and we say that nail is contaminated with bacterial cells. So the bacterial cells in my diagram are shown here in green. So these little green bacilli, these would be bacteria entering the skin. And of course, this is a breach of the first line of defense, skin. So bacteria get in through this very important barrier. And once they breach that first line of defense, the next line of defense comes to our protection. And recall what happens when the bacteria are present in this damaged tissue. So inflammation gets activated. And if you recall, there are cells, skin cells that have become damaged in this puncture wound, and these cells, these damaged cells, they begin to release cytokines. They begin to release these inflammatory signals. We'll show these as little blue dots. So these injured cells, they release inflammatory signals, cytokines, and then those signals, they bind to this cell here, this purple cell, purposely drawn purple because that's how it appears in a histology stain, where we see a lobed nucleus here, and we see many purple granules in the cytoplasm. So what cell is that in purple? You remember? Yes, it's in the tissues. So this is a mast cell. And so that mast cell degranulates and releases these little purple granules. And if you recall, these are histamine. And so histamine initiates vasodilation. So histamine comes along and binds to these endothelial cells lining a blood vessel. So this is a blood vessel. So we have histamine release initiating vasodilation. So the blood vessel vasodilates. Remember what that means? It means that the blood vessel wall becomes leaky. In other words, there are gaps that form in between the endothelial cells. So I've shown all these red cells as endothelial cells, and I purposely spaced them out one from the other so that you see the vasodilation. So we see that there are these gaps that form in between the blood vessel cells. We call that vasodilation. And remember what that does. Well, it allows fluid to leak out of the blood vessels. So they become leaky. We get fluid loss out of the blood vessels. And the purpose of that is to allow for white blood cells to squeeze out of the blood vessels and enter the tissues where they can perform phagocytosis. So remember how that works. So after vasodilation, these little sticky proteins form on the surface of endothelial cells, and we call those selectins. So the selectins form, and the selectins are like sticky little Velcro proteins that bind to the surface of these phagocytes. So I've shown a couple phagocytes that you should be aware of. Actually, how about this one in pink? This guy on the far left, and with a purple nucleus, multi-lobed, and then a pink stained cytoplasm. Do you remember who that is? Based on our histology lecture we did. Okay, so yeah, that's a neutrophil. So that's a neutrophil. And then over here, I've got another white blood cell stuck between two selectins. And in the blood, it is very large and it's an inactive form. Remember, this would be a monocyte. And so do you remember what we call when they stick to selectins? So when they're sticking to selectins, so in this example, they are sticking to selectins. We call that margination. Okay, so they stick to the selectin proteins. That's because the flow of blood is very, very fast through the blood vessels, and you want to make sure that you grab onto those particular phagocytes that we want. So they grab on, and then what happens? Well, as shown in this picture here, if you can see what I'm doing here, I'm circling this guy squeezing out. So this actually becomes an activated monocyte. Remember what monocytes are called when they activate? Yes, they're called macrophages. So the monocyte activates to a macrophage, and it squeezes out. And as it squeezes out, we call that process diapedesis. So they squeeze out of the blood vessels and into the tissues. And then they do their job. What's their job? Phagocytosis. So these are phagocytes. So I drew a macrophage. Remember, this is a symbol macrophage as well. So a macrophage digesting the green bacterial pathogens by phagocytosis. We studied that process in detail in a previous lecture. And then it basically chomps down on the bacteria intracellularly, releases the little bits of it by exocytosis. And then over here, I've got another guy here. Who's this guy? So we've got another white blood cell leukocyte oozing out, or it's going through diapedesis here. It's squeezing out through these little gaps that have formed in the blood vessel. And he's got all these these real long finger-like projections. Who is that? Yes, that is a dendritic cell. also a phagocyte. So we're seeing our key phagocytes in this story. We're seeing the neutrophil, we're seeing the macrophage, and we're seeing the dendritic cell. Those are our big phagocytes, our three big ones that play a major role in removing pathogens that have invaded the body by phagocytosis. Okay, so that's a review of the inflammation lecture. So now I want to talk to you about complement. So now that you see these two mechanisms of second line of defense all in one slide, so we're looking at inflammation and we're looking at phagocytosis all in one. You sort of see how that all connects, hopefully. So complement, if you think of something that could help or assist or really even amplify this process to make sure all of the steps occur correctly, that would be the job of complement. Now, if you notice off to the side, I have a picture of a liver here. Well, to the best of my ability there. The liver organ. And that's because this is where complement is, where complement proteins are produced. So the liver releases these proteins, and we call them complement proteins. Let's just make a circle with a C, okay, to represent that as a complement protein. And then these complement proteins, they become activated. And why do they become activated? Well, one thing you need to remember about this whole story we have pictured here is that there is a cascade reaction of what's happening. In other words, it's multi-step. But also, these phagocytes, do you remember that they secrete alarm signals? Do you remember that? So this macrophage would be eating the pathogen, but it also would start releasing a chemical alarm signal called an interleukin. Remember, interleukins are a type of cytokine. So these interleukins get released by phagocytes. And interleukins have various jobs. Might as well go ahead and throw in that interleukins initiate fever. So we have talked about fever in previous lecture videos. So how does the brain know to increase body temperature? Well, it gets a signal from the macrophage, the interleukin signal. So it signals to the brain, initiating fever response. But interleukins, they also go to other white blood cells. So other white blood cells are receiving this message, and it's going into the bloodstream. These little chemical interleukins are going into the bloodstream, and it's recruiting more phagocytes as well. It's like sending an alarm bell to the troops and saying, we've got an invader and there's only a few of us. I need to call for backup. I guess more like calling for backup. The police show up at the scene, and maybe there's only a couple officers that show up to a crime scene or an active crime scene. And then those police officers, what do? Well, they get on their walkie-talkies and they call for backup, right? So that's what's happening here. These phagocytes that are in the process of digesting the bacteria in our story, well, they're going to send a message to other phagocytes and ask for backup. Okay, so that's also happening. And then these interleukins will end up in the blood and they'll end up traveling to the liver. And then these complement proteins start being released. And it's actually a complex series of events for complement to become activated because there's all these enzymes that cleave the complement proteins into various pieces. And if you happen to find another video on it, I usually don't like to show you other videos on complement because everybody gets all panicked as they see how complicated it can get with all these different enzymes and So let's just simplify it and just say that there's an interleukin signal that reaches the liver, and this is a signal that tells the liver to begin producing and releasing complement proteins that become activated. Okay, so there are 3 important things to remember about how complement works, or what complement does. And the first thing it does is it amplifies inflammation. So it's going to increase inflammation. That's what amplify means. Okay, so how does it do that? Well, it does that by entering the blood and then binding to the surface of this cell, this purple cell. Do you remember who this purple cell is? Let's take a look at how I was trying to draw it. So purple multi-lobed nucleus, mini purple granules in the cytoplasm, resides in the blood. Oh yeah, it's a cousin of the mast cell. Basically, mast cells live in the tissues, and you remember this guy? Who's this guy? Yeah, basophil. Basophils live in the blood or migrate through the blood. So complement binds to basophils. Well, what do basophils do? Same thing the mast cells do, degranulate and release histamine. So the basophil will then release the histamine. So we'll show these purple granules coming out of the cell of the basophil. So remember, the histamine is what triggers vasodilation and triggers selectin proteins to form these sticky proteins. So it's going to enhance the signal that's already being sent by the mast cell. Okay, sort of like if the mast cell is ringing a bell, and that bell is essentially the histamine that's alerting the body and starting this mechanism in action. You have this bell ringing, and then you get the basophil activated in the blood, and now the basophil starts ringing a bell, and now we got 2 bells ringing, and we've amplified the signal. We've increased the strength of the signal by having multiple cells ringing the same bell, if that makes sense. So activate or amplify the inflammatory response is one job of the complement proteins, and they simply do that by binding to the surface of basophils. And when basophils get bound by complement, this triggers a chain reaction inside the cell that tells the cell to activate and to release histamine. All right, so what's the second thing? The second thing that complement does? Well, the second thing it does is actually assist with phagocytosis. So amplifies phagocytosis. Remember, that's the other big thing that we want to have happen here. We want those bacterial pathogens to get digested by our macrophage. And how's that going to happen? Well, it's going to happen if we have a lot of phagocytes at the scene. And it's going to happen if those phagocytes are aware of what they should be digesting, the bacterial pathogen. So the complement proteins, not only do they circulate in the blood and bind to basophils, but remember, the blood vessel is super leaky right now, meaning everybody's leaking out into the tissues. So complement proteins can leak out into the tissue. So imagine some of these complement proteins leaking into the tissues. And then what they do is they actually they bind to pathogens. They bind to the bacterial cell, to the surface of the bacterial cell. You see that up here in the right corner? So we've got the complement protein bound to this little green bacillus. This is a bacterial cell bound with complement. And we call that process, we actually have a word for it. It's called opsonization. And opsonization means to coat the bacterial cell with complement proteins. And then what that allows is there's a binding site on all phagocytes, where phagocytes have a complement receptor where they can bind to complement. And when that, in this picture I'm showing this dendritic cell here, he's ready to be called into action. He's ready to get in there and start eating bacteria. But he's sort of moving around in the tissue right now. And if his complement receptor here in black, this is a complement receptor. If this happens to hit a complement protein, that initiates a chain reaction inside of the dendritic cell to say, oh, okay, let me eat that. And so it then engulfs the bacteria. So that can oftentimes speed up the process of phagocytosis. So usually, remember, phagocytes bind via a pathogen recognition receptor to a pathogen, but it has to have that particular receptor that is in the right orientation to bind to an antigen on the pathogen. But if complement is there, sometimes that'll speed up the process of phagocytosis. Okay, so this slide is getting kind of busy. Let's go to the next slide. And in this picture, we're gonna describe the third thing that complement does. So let's summarize what we've learned so far in this video on complement. Remember, it's complement with an E, and it means that it's going to be helping. So these are helpers in the second line of defense. Their job is to make sure the events of the second line of defense are happening and to amplify the signal to make that happen. Inflammation and phagocytosis specifically. So we said, number one, it's going to amplify inflammation. Remember, inflammation is already happening, but it's going to amplify inflammation. And then how does it do that? Make sure you understand the how. How is it doing that? Well, it's doing that by binding. Let's go ahead and draw in a basophil here real quick. So this would be a basophil. So the complement proteins will circulate in the blood. They will find a basophil. They will coat the surface of the basophil, and that initiates a chain reaction inside of the basophil. So the basophil will degranulate. So degranulate, which means it will release histamine. And histamine has all these effects within inflammation. So histamine is going to be triggering the vasodilation, and histamine is going to signal selectin formation. So those little sticky proteins are going to start forming as a result of information from histamine. And then histamine is also going to signal the phagocytes. So it sends a signal that binds to phagocytes and attracts the phagocytes. The phagocytes are attracted to histamine. Okay, so they hear this alarm bell, and they're designed to go to where that alarm bell is ringing. And so they're able to sense histamine as a chemical attractant. So although this is already happening due to the mast cells are doing this, we said it's like the basophil is ringing another bell and amplifying or increasing this response process. Okay, the second part of complement we said was to amplify or increase phagocytosis. So that's the other important thing we want to have happen. And how? How does it do it? Well, remember these bacteria. So this green bacterial cell is in the tissues and it's up to no good, okay? So we want to eliminate him by phagocytosis. So to make that happen faster than it would normally happen, complement binds to the surface of the bacteria and then that's beneficial because phagocytes have complement receptors. So a phagocyte has complement receptors here that bind to complement. Receptors are really important to understand in the immune system. Lots of cells have these proteins on their surface with a particular shape, okay, like a lock, and then there's something that fits in it like a key. That's the concept of how receptors bind to things, bind to other molecules. It's like a lock and key. And so when this particular receptor, the lock, comes in contact with the complement protein, which is the key, this is the magical reaction that triggers the phagocyte to say, okay, I'm going to eat this. This is a foreign bacteria that I need to digest by phagocytosis. And remember, we call that process opsonization. I like to think of opsonization like candy coating. Think of it as though the bacteria is candy coated by complement. And so we want the phagocyte to eat the bacteria, but if you coat the bacteria in complement, that's like candy coating the bacteria for the phagocyte. The phagocyte is more likely to quickly process or digest this bacteria. Okay? It's like if you want your kids to eat something, it's got it, if it was covered in candy coating or covered in sugar, that's how I get my kids to take their vitamins. Boy, those vitamins they take, they taste pretty good. Taste like candies, don't they? Those gummies. But, it provides a benefit for them to take the vitamin. And they're more likely to eat the vitamin, the gummy vitamins, because it tastes like candy, as opposed to if they ate a vitamin like I take that has no taste to it, they're less likely to eat that, right? They're more likely to eat the candy coating. It's the same thing with the phagocytes. They're going to be more likely to digest the pathogen if it's coated in complement. We call that opsonization, to increase phagocytosis by binding complement to the pathogen. Okay, third thing that complement does. So the third thing that it facilitates is what this figure is trying to show us, and it's called cytolysis, which literally means cell or cell lysis, cytolysis. It means to destroy the cell membrane of pathogens. So in this picture, they're showing us all the complement components, and really we don't just write it as C. There's multiple types of complement proteins, but don't worry about all the numbers associated with them. But the important part is to see that there's actually this pore. There's a pore that forms made out of complement, and this pore actually causes a hole to form in the membrane of pathogens. Let's go to the next slide here. This is called the membrane attack complex, which is sometimes abbreviated MAC, but this stands for membrane attack complex, or the MAC. What that means is that this collection of complement proteins, they're organizing together and they're forming a hole in the bacterial cell wall, particularly gram-negatives because they have that outer membrane. And that's what we're seeing in this picture. Whoa. In this picture, we're seeing a big hole. Do you see the hole? So this hole forms out of complement proteins. What happens if you have a big hole in your cell wall? Well, yeah, fluid rushes in. So we get water entering the cell rapidly, and the cell will expand and burst. We call that cytolysis. So the cell, and then usually this is gram-negative. We're talking a gram-negative. Bacteria is, for example, would be the target. So the gram-negative bacteria gets a big hole in its wall. That's not a good thing for the bacteria, but a good thing for us because it's going to kill the bacteria. So we get cell death, or what we call cytolysis. So those are the three events that are caused by complement, and that wraps up actually our discussion of non-specific immunity. So in the next lectures, we'll talk about specific immunity. And I'll talk to you next time.